Thank you very. Got it. Okay. So you can hear me okay, see me okay, everything's all right. Great. Well, I'm very happy to be here for Build-A-Cell. So thank you to Carlise and thank you to Kate out of Mallow for the invitation. I guess I was told I should introduce myself. So my name's Dan Hammer. I'm a professor at the University of Pennsylvania. I have joint appointments in chemical engineering and in bioengineering. I grew up in New York City, as we were just talking about. I went to high school at a place called Stuyvesant, undergraduate at Princeton, graduate school at Penn, where I am now. I spent 8 years at Cornell in Ithaca in chemical engineering, and then came to Penn in 1996. And also, I was recently named the director of the Center for Precision Engineering for Health with CPE4H at the University of Pennsylvania, which is a $100 million hiring initiative at the university. And these are the ways to contact me: email, web address, hammerlab.com, and I'm on Twitter at Professor D.A. Hammer. And what I'm going to talk about today are synthetic motile cells and designer membrane organelles. And I've had two amazing collaborators in this work, and I want to mention them right up front. Matt Good, who's in cell and developmental biology at Penn, who just got tenure there. He's an expert at making membraneless organelles through coactivating proteins. And Daeun Lee, who is a chemical engineering faculty member who's an expert in microfluidic assembly. And the three of us have been working on various projects related to synthetic cell design over the past 6 or 7 years or so. And the work I want to talk about is funded by the DOE, the NSF, and the NIH. So it's great to it's great to give a talk like this. Oh, I want to advance. How do I advance slides? Oh, okay, so I have to learn how to advance slides. So it's great to give a talk like this because I can go back in time and tell you the whole history of everything. And sometimes it's really great to hear the history of things because you wonder how someone gets from one place to another. And for me, it's a particularly interesting story because I hate working on the same problem for too long. I have a very short attention span, and I want to go from problem to problem, make some advance, and then move on to something else that piques my curiosity. So our adventures in synthetic cell design emerged from what I did on my PhD thesis, where we studied cell adhesion under flow. And this is a classic cascade that I'm illustrating here for the process by which cells get out of the bloodstream and move into a tissue. So for example, if you cut yourself and bacteria enter your tissue, there's a certain cell type that you have in your circulation. It's called a neutrophil. It usually is whizzing by in the bloodstream, but what happens is the tissue upregulates adhesion molecules that act as a molecular flypaper. They capture the cell. The cell first rolls over the surface, and then it comes to a stop, then it changes shape, and then it transmigrates and moves into tissue, and then it hunts down whatever invaders have entered your body. And the key molecule that mediates rolling is this molecule here. It's called a selectin. Selectin stands for selective lectin, and a lectin is a protein that binds to a carbohydrate. And the carbohydrate that selectin recognizes on the surface of the leukocyte is called Siluous X. It's a particularly structured pentasaccharide on the surface of leukocytes. And to give you an idea of what this rolling interaction looks like, I have to go back to not using, what am I doing? Okay. I'm not trying to highlight, I'm trying to just get rid of the so I'm trying to just click this slide to get it to run. Oh, oh, there you go. Okay, so what what is, that was such a mess up. So what you're seeing here in this slide are leukocytes that are rolling along the vascular surface. The way this image was taken is in the mouse cremaster muscle. And what's happening is that my collaborator, Ingrid Sorelius, she just pushed her objective against the muscle of the mouse cremaster muscle with, and then initiated an inflammatory response. And what happens is the leukocytes, which are shown in white, they start rolling along the blood vessel wall, and that's dictated by a certain chemistry. The dark streaming down the center line of the image are the red cells. They're whizzing by, but the molecular flypaper is slowing the leukocytes down so that they'll roll over the surface. So what we did in our lab was we recreated this in what is a cell-free system. This is our first synthetic cell in the Hammer lab, if you would. This is leukocyte rolling in a cell-free system. So Deborah Brunk, the student who did this, she basically, I'll go back and show this again. She basically put the carbohydrate cellulose X on the surface of the microspheres and then made a surface of one of these selectin molecules, E-selectin. And you could see you could recreate this rolling response just by putting the right molecules on the surface of these microspheres. And this body of work, which I'm not going to talk about any longer, I'm just telling you how I got involved in synthetic cell work. This body of work has led to lots of different technologies that various people from my lab have taken on. So for example, some of you may know a woman named Lola Anayola from University of Michigan. What she did was she took this idea and then built porous microspheres that had the adhesive properties of leukocytes and loaded them with drug and was able to deliver them to inflammatory sites in vivo. So there's lots of different ways you can use this synthetic cell technology for things like drug delivery. But I'm not going to talk about that anymore. So what I am going to talk about is our stepwise progression in trying to make protocells that can do various things. And this is all summarized in a review paper that I wrote with Nea Kamat, who's at Northwestern, and Josh Katz about 10 years ago. And so the idea behind protocell design in our minds is to try to build something that has the structure of a cell, a plasma membrane, organelles, but is made out of designer components that aren't usually used in cell design, where you can build in advanced functionalities into those components that you can't otherwise accomplish in nature. A simple example is in the plasma membrane. So the plasma membrane of a typical cell is a phospholipid bilayer. And we all know about phospholipids. They're wonderful molecules. They oxidize very quickly. They're tough to work with. They're difficult to manufacture and modify. But what if you could make membranes out of other things? What if you could make them out of, say, polymers or proteins? Then you could use the molecular toolbox of polymers and proteins in order to build functionality into these membranes. And then the next step, and what I'll show later on in the talk, is how do we build organelles into these structures? And the basis of what we're going to try to do is build membraneless organelles that are made of coacervating proteins that act as hub of biochemical reactions that we can tailor so we can drive certain pathways inside of these protocells. And then what we're ultimately interested in is actuation. So how do we get these protocells to move from place to place? How do we get them to crawl over a surface? How do we get them to secrete on cue? Those are the kinds of long-term goals that we have for these materials. And I'll touch upon all of these different ideas in the talk today. So let me just start with the idea of the membrane. So, and this allows me to talk about another historical fact in my earlier career. And so we know about membranes, we know about vesicles, we know about the typical phospholipid bilayer. Typical membrane of a cell is a phospholipid bilayer, diacetyl chain phospholipids. They orient in the lamellar phase, and they can be an enclosed structure. And depending on the diameter of the structure, they will either be giant vesicles if they're a few microns. They could be small vesicles if they're 100 nanometers or so. So I was chatting a bit at the beginning about Frank Bates, who is a faculty member in chemical engineering at University of Minnesota. And I saw him give a talk at the 1994 APS meeting. I think it was in St. Louis. And he was talking about polymers that he had synthesized that would form the lamellar phase. In this particular case, he had done the polymers in the melt. And I went up to him afterwards. I said, well, if you could make polymers that form a lamellar phase, we could probably make them into vesicles. And he said, that's a great idea. Let me think about that. At the time, because he'd only done things in the melt, he was concerned that the solvent effects would affect the ability to form a lamellar phase. And then when I moved to Penn, we were involved in the MRSEC. We got a seed grant from the NSF through the MRSEC, and Dennis Disher and I and a guy named in physics named Phil Nelson, we were on the seed. And I said, you know, I had this idea that we could make membranes out of polymers, and let's talk to Frank Bates and see if he has any polymers that are suitable for this. And at the time, he had worked out the phase diagram for diblock copolymers in aqueous solutions. And he said, I have just the polymer for you. He sent it to us, and then within a week, we had polymer vesicles. And so many of you know the polymer vesicle story. I mean, polymersomes were invented through this collaboration. I should say that there was work going on in A.D. Eisenberg's lab at the time where he had taken various different polymers and different solvents and shown that you could get closed cell vesicle-like structures. But the focus we were making is to try to do something biological, making closed cell structures in an aqueous solution. So it's not as if there was no work on closed cell structures before we got started, but I wasn't aware of the Eisenberg work when we came up with this idea. But as far as making vesicles in aqueous solution, this works really great. So this is the original polymer that we use, a polyethylene oxide polyethylethylene. There's a closely related family of polymers, polyethylene oxide polybutadiene, that work really well as well. The polymer is much, much bigger than a phospholipid, and that confers properties of stability into the membrane that you don't have with phospholipid membranes. So I don't know if I need to summarize the whole world of polymersomes, but as you probably know, there are thousands and thousands of papers on polymersomes that have been written now, different kinds of polymer architectures, different chemistries, different stability, different functionalization. It's a very popular area of research for making synthetic cells or capsule-like systems. So what I want to talk about in this talk is using polymersomes to make a motile cell. There's lots of work that's gone on in self-motile cellular-like structures. Some of you probably work on this. There's been a lot of work on microswimmers, which are poles of platinum and gold, which conduct an oxidation and reduction reaction pair that drives a water flux that causes the swimmers to move around. That's like a bacterial-like motion. But what we've been enamored with is the idea of using enzymes to generate force. And this is an idea that originally, I think, came from Ayusman Sen, who's at Penn State. And the basic idea is that when an enzyme turns over a substrate into a product, it generates a little bit of a force. There's a little bit of an impulse that acts on the enzyme. There's a lot of debate about where the impulse actually comes from. Is it exothermic interactions? Is it that there's an osmotic influx to the molecule that gives you the water gives it a momentum push on the enzyme? Lots of debate about the actual fundamentals of how the force is generated. But nevertheless, it's very clear that when you put enzymes on a material and feed it substrate, you can generate forces and generate motion. Ayusman demonstrated this just in a simple experiment where he took a sphere and coated it with urease and then added the substrate urea. And then the sphere would move around faster. And you could back calculate from using Brownian dynamic simulations that the force that generated per turnover event was about 10 piconewtons. And that gave me an idea, which is to say, because most biological adhesion molecules break at about 100, 200 piconewtons of force. And so what I wanted to do was make an adherent polymer vesicle and put it on a surface and get it to crawl around on a substrate. Very much akin to what I'm going to show here in this, what I can show here in this, I still can't get this pen. I don't even know what I did here, but I wish I could undo it. Oh, there you go. Okay, I figured it out. Okay, so very much akin to the way these macrophages crawl over surfaces. So the other side of my lab works on real cell motility, and so this is an image done by Laurel Hine, who's was a graduate student in my lab, is now on the faculty at the University of Colorado. So real cells, when they crawl on surfaces, they adhere to the surface and they extend and then they contract and then they pull themselves along. So we want to make a motile system that behaves somewhat like this. So this is a collaboration with Dayan Lee, and the postdoc who did this was Wushik Zhang, who's shown here in the bottom left. And the idea was pretty simple. So we took polymer vesicles that were made by self-assembly, just rehydration in this case, and we loaded them with an enzyme called catalase. So catalase is in the interior. And then we adhered those vesicles weakly to surfaces using biotin-avidin interactions. Now, biotin and avidin are strong molecular bonds, but we used a very sparse density of them so that the overall adhesion turned out to be relatively weak. The catalase in these experiments actually migrated to the membrane, so we could actually see it on the surface of the vesicle. So we know the catalase was membrane associated in these experiments. And the catalase converts hydrogen peroxide to water and oxygen radicals. So we just fed the solution hydrogen peroxide, it goes across the membrane, it's converted by catalase, and a force is generated and the particles are motile. So here's what this actually looks like in very sped up fashion. So these are particles that are adhering to a surface. They're fed hydrogen peroxide and they start moving around. They start translocating over the surface. So they're motile on the surface. And we could measure the mean square displacement as a function of time. And it's diffusive. It's in biology, that's called a random motility. So we generated this random motility of particles on the surface by feeding the substrate to the enzyme. And so the various different ways that we can look at this. First of all, this is a slowed down version of a video that shows the motion. You can see that the particles are jiggling around on the surface. So they're translocating. All of the particles in the field of view are translocating. Just to show you a little bit about what, I'm gonna go back, show you in more detail what's going on. Notice this little fleck of dust here. This is in real time, where we're watching the motion of the particle, and you can see that it's moving in little discrete jumps. So what's happening on short time scales is the particles experiencing this force, and that's causing bonds to break. And the bonds that are breaking, they break and then they reform because there's such a sparse density of them. So the adhesion, tuning the adhesion in this experiment is very critical to get it to work the right way. But by the time this little video is done, you see that the particle is translated very far away from this little reference speck that we have in this video. And the motion depends on the amount of fuel you use. So if we use a low concentration of hydrogen peroxide, as shown here at the top, we get modest motion. But if we use a high concentration of hydrogen peroxide, we get a lot of motion. So it's definitely dependent on the enzyme and the substrate. And one way to look at that is on the graph. If we look at the diffusivity or the random motility of the motion, you see that as you increase the concentration of hydrogen peroxide, you get increasing diffusivity. And this is on a log scale. So these are substantial changes in the diffusivity. And of course, by leaving hydrogen peroxide out, you get the negative control. There's no motion of the particle in the absence of fuel. Also, the experiment can run down. So what can happen over time is that you exhaust the fuel, and when you exhaust the fuel, the particles stop moving. And so in depleted, this isn't very interesting to see. When the fuel is depleted, the particles just sit there. But you can reinitiate the motion by adding fuel back in, and then the particles start moving around again. So the motility can be reactivated as the fuel is added. And then finally, in everything I've shown so far with this experiment, everything is uniform. There's no gradient. There's no directional motion. The particles are they don't have a polarity. They're put in a uniform field of the fuel, and they move at random. But you can get directional motion with this. And so the last thing Rushik did before he left was he made a gradient chamber and made a gradient of hydrogen peroxide. And so, and in that gradient, which he could characterize by solving the reaction diffusion equation, he could show that in the absence of adhesion, the particles flew toward the source. That's what's shown here in the center panel. But if you add adhesion to the substrate, what happens is the particles migrate to the source in a very orderly way, because there's this enhanced friction on the substrate because the molecules the adhesion molecules are slowing the particle down. So, so there's lots of open questions with this experiment, but the one thing we wanted to, different enzymes, different directional motion, but the one thing we wanted to address is, could we make the particles uniform? And that's what Dayon and I have been working on recently. And the student who's done this, a wonderful student named Jess O'Callaghan, who's shown here at the bottom. And so some of you may know that the Lee Lab is the world's expert at microfluidic assembly. And this idea is that you could take an aqueous solution, inject it into an oil phase, and that goes into an aqueous depot. And you basically make water and oil and water double emulsions this way. And this is a wonderful way to make a very large population of uniform cells at high density, a uniform cell-like structures at high density. In this particular case, we didn't use polymersomes, we were using protein shells. So we use polylactic glycolic acid to make the shell structure. And you can see that we get these nice uniform particles that are labeled with a fluorescent dye in the shell. And we can do various things that I'm not going to talk about changing the shape of the shell, compressing it down by a technique called osmotic annealing. We can make a variety of different structures, but these are really large particles. I mean, they're 50, 60 microns to start with, and then we can squeeze them down to 20 microns or so. So now, and Jess has been using the enzyme urease to watch the motion of these particles. Now, first, I'm going to show you what happens with single particles. So if we have single particles, this left-hand panel here is just the control experiment. There's a little bit of drift in the experiment in the absence of the substrate, which is in this case is urea for urease. But if we add urea, we get substantial motion of these particles over time. Now, if we make the particles imperfectly, meaning that if the particles aren't uniform and monodispersed, but they come in dimers and clusters of various kinds, we see some very interesting motions of these particles. So in what you're about to see, the red traces indicate what's happening to single capsules, and the blue traces indicate what's happening to clusters. So what you'll see is that the clusters move around in all sorts of different kinds of trajectories because you've broken the symmetry of the particle, and because of perhaps clustering of the enzyme together, and certainly because of hydrodynamic interactions, you get all these very, very large expansive motions of these particles. By the way, in these experiments with these that I've been talking about in the last two slides, the particles are in solution, they're not adherent to a surface. So, one of the big issues now is to get these particles adherent to a surface, recreate the experiment that I did before on adhesion. But what's very interesting to us is that if you have some kind of multi-particle cluster or symmetry breaking, you get some very interesting motions to the capsules that we're trying to understand. And of course, if you just look at the mean square displacement, you get much bigger mean square displacement of the clusters than you get of single capsules. Okay, so that's work in progress. We haven't published that yet, but it shows you a bit of what we're working on currently with enzyme-driven motion. So what I've talked about so far is making membranes out of polymers, and that leads to another feature of our lab that I wanted to talk about, which is we've generally been interested in the evolution of protocells, the evolution of vesicles, different ways to make membranes. And so we know about lipids and we know about making them with polymers. They all have their advantages and disadvantages, but there's a lot of evidence that you can make membranes out of peptides from Tim Deming's lab. And that led us to an idea that maybe you could make membranes purely out of proteins. I mean, purely out of proteins. I'm not talking about putting proteins in a lipid membrane. I'm talking about making the membrane entirely out of proteins. And so the next step in the evolution is recombinant protein vesicles. And some of you may know that in biomaterial science, there's lots of reason why one wants to make membranes out of recombinant proteins. There's a question in the chat. I want to just check and see what it is. No. The question is, do clusters with a certain number of cells always make the same movements? And the answer to that is no. So, but what I should say is, what I should have said is, obviously what we're trying to do is catalog the kinds of motions you see with the kinds of clusters you get. And we're trying to actually orderly make clusters. So we're trying to make clusters of certain topologies in order to see what the correlation is. Right now, the clusters that we observe they're a happenstance. You know, we happen to make them, and then we see them move in a strange way. We're trying to be a little bit more deliberate about these experiments. Okay, so anyway, what I was saying is making membranes out of recombinant proteins. And so, you know, in biology, when proteins are made, they're made with great precision. You have a gene that codes for an RNA, RNA codes for a protein. The protein is always the same. You know, every single protein's got the same sequence, it's got the same size. And if you were to look at a MALDI of those proteins, you would see a single peak, as is shown here at the top. But if you have polymers, like are made for diblock copolymers for polymersomes, you get multiple peaks. So you can't avoid the polydispersity of making polymers. You know, you run a reaction, stop at a certain time, and you have a distribution of sizes. Some very good polymer synthetic chemists can narrow this distribution down quite a bit, have a very small polydispersity index, Frank being one of those people. But you can never get rid of the polydispersity. But you also have another advantage, which is that you can have complete control of the sequence of amino acids in these chains. So you have 20 canonical amino acids, you can put them in any sequence you want, which is the kind of control in polymer synthesis you don't have. And furthermore, for any biological applications, you have the advantage of putting in sequences that do something in biology. For example, you could put an RGD sequence in that allows for adhesion. You could put like a single chain antibody in that targets a receptor. You could put protease cleavable domains, like thrombin cleavable domains, in the center if you wanted to cut the protein at a certain time with a protease. So you can do all of these things with recombinant proteins. And so that's why we've been so keen on trying to make vesicles out of proteins. And so we have done this using a protein called oleosin. So oleosin is a naturally occurring molecule that occurs in plants. It's an amphiphile. It has a tri-block structure. It has an N and C terminal hydrophilic end, and then it has a central hairpin, which is hydrophobic. In nature, these proteins in this hairpin shape stabilize the fat globules in sunflower seeds and sesame seeds. So you've eaten oleosin before in your life, almost certainly. And a lot is known about the secondary structure of the hydrophobic core, and we know the sequence, and we have the gene, and we can code it. So the basic idea here is this is a good candidate for making a membrane because it's an amphiphile. It's a simple amphiphile, and we can modify it any way we want. So, so the student who did this was Kevin Bargo, who graduated some time ago, and everything I'm going to show you now is made by recombinant methods. So we get the DNA, we put it in E. coli, we produce a protein, purify it, and then do what we're going to do with the structures. And so the first simple thing to think about with oleosin is if we take the native structure of the protein, remember I said it's a tri-block, it's got an A and C groups are hydrophilic, the B core is hydrophobic, and the number of residues are 42 in the A block, 57 in the C block, 87 in the central hydrophobic core. We can just think about making a simple family of truncations. So not changing the sequence at all, but just lopping parts off. So for example, if I lop the entire A group off and just left 7 residues on the C block, I could have 0.877. And the nomenclature is obvious based on what we're doing. And so if you just take simple truncations of oleosin, you can make recombinant vesicles. So these are two examples of recombinant vesicles that have been made with two different proteins. They very, very clearly are vesicular shape. I'll show you demonstration that you can make giant vesicles with them. It's also interesting, you can make other structures if you're interested in other structures. For example, you could make like a worm like micelle. So if you take a protein that's highly asymmetric, 42877, it'll fold upon itself and it'll make these very long worms. So this is a worm that's hundreds of microns long in one dimension and then, nanometric in another dimension. Or you can make sheets. So the protein on the right is a protein that has very, very small head groups and then has a big hydrophobic core. And then that makes these uniform sheets on a surface. And so you can make these coatings on a surface if you want. Okay, so I'm going to go through this quickly. Just you get the central point. So we could show how you can modulate the formation of vesicles. We made a family that had 65 residues in the hydrophobic core and 4 different size head groups. And we just took those 4 proteins and we mapped out the phase diagram for their structures. So this axis here on the left is the hydrophilic fraction. This axis here is the ionic strength, meaning that if we have in full PBS, we're going to protect all the electrical charges and everything's going to be able to pack much better. In distilled water, we're not protecting the charges and everything will be repulsive and then they'll spread out. And you can get various different shapes depending on the size of the head group and the ionic strength of solution. So for example, everything at the upper right-hand corner of this slide is you make vesicles. So there are lots of different formulations, there are lots of different combinations where you can get vesicles to form. At the bottom left, you get sheets. At the top left, you get fibers to form. So even with the same 4 proteins in different solution, you could actually make different kinds of structures if you want. But the bottom line is you can make membranes out of proteins. And here's a wonderful picture of giant vesicles. So these are giant vesicles. This is this scale bar is 5 microns or so. So these are 15, 20 micron giant vesicles with calcine dye in the hydrophilic lumen, and then Nile red in the hydrophobic core. So we could actually see what the membrane is like. So that's a next step in the evolution of vesicles. And I know some of you work in the world of using cell-free synthesis inside of protocells. The idea being, you take all the machinery, DNA, RNA, ribosome material, and actually synthesize proteins in the interior of a protocell. But imagine if that protocell were made out of a protein. Then you could be making a protein inside of the protocell that could migrate to the interface and change the chemistry or change the structure of the membrane. Or you could do something like have a protein that's in a membrane that has a protease cleavable domain, and then you make a protease inside of your protocell, and then the protease would go out and would chomp the membrane protein in little bits, and you get this gradual secretion of material from the inside of the cell. That's not an exhaustive list of all the ideas you have, but once you can make a membrane out of a protein, you can do lots and lots of things with it. So that's what one of the things that we are trying to do. Okay, so I promised certain people that I would introduce a tachometer into this talk, and it turns out that I'm far behind where I wanted to be, and we're at slide 26, and we have 48 total slides. But don't worry. I won't go much beyond 45 minutes. So tachometer tells us where we're at the moment. So, okay, so back to this little figure. What I'm going to talk about for the rest of the time is our strategy for making organelles. And this is, again, in collaboration with Matt Good. And the basis of this is we're going to try to make organelles out of protein coacervates, membraneless organelles. And I know a lot of you are working on this or familiar with this area. So the basic idea is that there are these proteins that weakly interact with each other, and they find each other in solution. They phase separate into these liquid droplets that are known as coacervates. And it's not an aggregate; it's an entirely liquid phase of material, but they phase separate from the outer aqueous solution. And what we want to do with these coacervates is recruit enzymes and recruit surface molecules to them so that we can make functional organelles that can live inside our protocell materials. Also, by the way, I should point out that we also have a large effort in using coacervates in real cells. So we want to control cell behavior by making coacervates that sequester and release molecules inside of real cells. And we're doing a lot of that work too. I'm not going to talk about that today. Now, again, I want to emphasize, everything we make in this part of the talk, we're making recombinantly. So with precision synthesis of the molecules. And we got involved in this through the efforts of a wonderful postdoc, Ben Schuster. I think I have his picture on the next slide. And he wanted to re-engineer a protein called LAF1, which is a well-known protein discovered by the Brangman lab that sequesters and releases RNA on cue inside of cells during development. And the phase diagram for LAF1 is known well. Everything to the right of this boundary for LAF1 will coacervate. But the domain of LAF1 that's responsible for coacervation is the RGG domain. It's a 161 amino acid domain that has arginines and glycines in it, and it will coacervate by itself. So if you just isolate RGG, it'll coacervate, and here's the phase diagram for RGG. So what could we do with that? So this was Ben's idea. So Ben had the idea that 2 RGG domains are going to be better than 1. And if we make a dimer of RGG, we'll shift the phase diagram to lower concentrations or higher temperatures, depending on what the phase diagram is plotting. And if we put things like cleavable domains in between the RGG domains, we could reverse a dimer. We could take a dimer to a monomer, and then we can reverse the formation of the coacervate. So we could get coacervates to form. We could get coacervates to disappear. And then concomitantly, if we put different kinds of structures inside of these coacervates, we could get them to assemble together, and then we could get them to release on cue. That's the basis. That's the 0th order view of what we're trying to do with these coacervate systems. So if you take this protein that he made, so the first protein he made was an RGG dimer that had the thrombin cut site in the center. So a dimer. If you take that protein and put it in solution, it coacervates wonderfully at room temperature. And so this is a model membraneless organelle, if you will. And then we can demonstrate that, let's skip over this slide because of time. So, and then we can demonstrate that we could reduce the size of the droplets by using thrombin. So in this experiment, we take the protein, the dimer of RGG with the thrombin cut site in the center, and add thrombin. So it immediately coacervates, immediately forms these coacervates, and then the thrombin comes and cuts the dimer into monomers. And then at room temperature, monomers don't phase separate, and only dimers do, and then we reverse the phase separation. And that's indicated here by this phase diagram in the upper left-hand corner. The upper curve is the dimer phase diagram, and the lower curve is the monomer phase diagram. You can see that at room temperature, dimers will phase separate, monomers will not. So we have a reversible, not reversible, an irreversible system where when we cleave, we can get the dimer to fall apart. And this actually works with other proteases. Of course, you don't need just thrombin. You could use a TEV cut site and then use TEV as a protease, and this works fine. You can modularly insert any kind of protease that you want here. So now could you get organelles to form on cue? And the answer is yes. And so the strategy that Ben used here was he attached the dimer of RGG to a very large protein called the maltose binding protein, which prevents coacervation. It's a solubility tag, if you will. And he put the 3C cut site in between the maltose binding protein and the dimer. And then if you introduce the 3C protease, it cuts off the maltose binding protein, you liberate the dimer, and then you get the formation of membraneless organelles. So you can form organelles on cue as well. And we took that to the next level by using light as a way of inducing coacervate formation. So this was done with a protein known as focal. We didn't invent focal, but there's a published report that's shown below, Zhang and colleagues, where this is a coated protein that you can put inside of a molecule, and it irreversibly cleaves at a wavelength of 405 nanometer length. So, so what we did with that was shown here. This experiment was done by Ellen Reed, who's currently a postdoc in the Tucher lab at Princeton. So it takes a little while to explain what this experiment is, but here's the RGG dimer with an M-scarlet reporter. That molecule by itself, RGG, M-scarlet, RGG, would phase separate if it were alone in solution. But what she did was she appended this very large MBP on the end, which prevents coactivation. It's a solubility tag. And the intervening molecule is focal. So if you illuminate this molecule with 405 nanometer light, you cleave the focal, the MBP comes off, the RGG dimer is liberated, and then you get organelles to form on cue. And that's what's shown here. The upper row is before illumination, and the bottom row is after illumination. So you get these coacervates to form on cue inside of your material. So what are the kinds of things that we're hoping to do with these coacervate systems? Well, we want to control biochemical responses inside of protocells by assembling the right molecules together. So one example is we could take split enzymes, and I'll show you an example with N and C on Nanoluke, where we could put them in between two RGG domains, and when they're assembled together, they're functional. The second is that we could use the organelles for compartmentalization of cascades of reactions. So we could have, let's say we have enzymes X, Y, and Z. We can co-sequester them all together and make a functional organelle that where the substrate of X ultimately becomes the product of Z in this tight environment. Very much the way organelles work. That's the way, say, mitochondria works by working as a hub of various different enzymes. Well, we could do this in an inducible way where we already self-assemble X and Y molecules together, and then we add a small molecule that allows the Y molecule to come in. And then in response to the stimulus, we can get this cascade of reactions. And I emphasize again, all the condensate work is done in collaboration with Matt Good. And these are ideas that he and I have been pursuing. So just to demonstrate the feasibility of this is a work that was done by Mu-Yang Guan. I think I have a picture of him on the next slide as well. And he was able to sequester a light-emitting enzyme inside of coacervates known as nanoluciferase, or Nanoluke. It's a very bright version of luciferase, the same enzyme that you find in the firefly tail. And we could get very bright light emission from these droplets this way. We could also modulate the amount of nanoluke that was in the droplet by blending a nanoluke containing RGG with a non-nanoluke containing RGG, and then we can modulate the amount of light output from these materials. And then separately, we could show that we could induce with a trigger the assembly of an enzyme inside of the coacervate using the strategy I talked about before, where we add an agent and then it induces the self-assembly in the coacervate. And this is done with a split version of Nanoluke. Nanoluke can be split into two bits, large bit and small bit. It's all coded by molecular biology. The affinity for the binding between large bit and small bit is not very high. So it's in, at the point, it's in the hundreds of micromolar range for KD. So it's not a very strong association, but if you co-sequester them together in the same coacervate, then you're going to get a large amount of assembly and a large amount of light that's emitted. So the idea behind this experiment is we put the large bit in between an RGD dimer, and then the small bit, we use the same strategy I just showed before, RGG, small bit RGG, appended to a maltose binding protein that prevents it from going into the coacervate with a cut, in this case, a cut site, 3C cut site. So we add 3C, that cuts off the MBP, the RGG, small bit RGG finds the coacervate, they co-assemble, and the nanoluciferase becomes active. And that's what's shown here in the case where we actually do the experiment the way I described. We get the assembly of large bit and small bit together, and they emit light. So we can actually trigger enzymatic activity in response to a protease. Okay, so what are some of the other things that we could do with these coacervates? So we can use other proteins in order to assemble molecules into coacervates. I think of these proteins as little molecular Legos. the synzips, which are synthetic leucine zippers, which were developed by the Keating lab, we've been using the pairs synzip1 and synzip2. We also have work that's been going on with sets of molecules that come from Roman Giralia's lab in Slovenia. He's made a pair of 6 pairs of peptides that allow for self-assembly, and some of those seem to work pretty well. And so what we can do in summary with all these is that we can put cargo inside of droplets. I just showed an example of where we put nanoluciferase as a cargo inside of RGG. In this case, this is a fluorophore, just as a demonstration. It'll go in strongly into the droplets. Or we can connect cargo to a droplet using synzip domains, synzip1 binding to synzip2. Here's an illustration that we can use fluorophores and get them into droplets this way. And then we can release things from the droplets. So we could take a cargo that's in a droplet and cut it out with a protease, or we can have a cargo that's attached to a synzip that we appended to the surface and then release it, and then we can show that we can release those molecules. So this is a wonderful strategy for either for cells or for protocells to have a cargo inside of a droplet, and then you have some release over time by either a protease or light. I demonstrated light as a way of doing this as well. Now, finally, I know I'm over time, so it'll just be 1 or 2 more minutes. So finally, we figured out a way that we could put molecules on the surfaces of coacervates. That's by making a surfactant coacervate. The idea is you take an RGD molecule that inserts into the coacervate and then have a GST domain, which is soluble, won't go in, and that's going to migrate to the interface of the coacervate. So here's a demonstration of a red fluorescent protein migrating to the surface of a coacervate. And we've been able to use that strategy to put active enzyme on the surface of a coacervate. We used catalase HP2, which we can code by molecular biology. We attached it by synzips to the surface of a droplet. The green fluorescent protein reports where the catalase is. And we can then get these little coacervates to jiggle around in solution in response to the action of the substrate through the binding and activity of the catalase. So we could put a variety of different enzymes and other structures on the surface of the coacervates at will using the synzip strategy or others. So that's the end of my talk, I believe. So what I'd like to finish by saying, other than showing you a picture of my lab, is that our ongoing work is to continue to work on protocell motility, effective gradients. We want to measure the motion of protocells on micropad arrays to measure the forces of migration, use different enzymes, look at the collective dynamics of particles as they move around. We have further work going on in organelle design, genetic encoding, building in active enzyme cascades, integrate different enzymes in the same organelle. And then finally, we're interested in combining technologies where we have activating enzymes and organelles that can drive the motion of coacervates from the inside out. So I thank you very much for your patience. You made it to the end. The talkometer says we're done, 48 slides. So thank you very much for your attention. I very much appreciate it.
Thank you, Dan. That was wonderful. I think we've got some questions that are rolling in the chat. We can keep addressing those. And then also if anybody would like to unmute themselves and ask, you can do that as well. Dan, I'll have you read the question out loud before you answer it, just for anybody who ends up watching the video later.
Okay, so there was the question I answered already. Do clusters with a certain number of cells always make the same movements? And the answer is no, but we're trying to catalog the motions. Next comment. Great talk. Thanks. Have you ever seen any limits to what types of proteins can be sequestered in RGD domain droplets, like size charge? Yes. So, and in the original Nature Communications paper, so there's two ways you could try to get a protein inside a coacervate. One is, does it naturally go in just based on size? And we showed very clearly that once you get to about 10K or so, you can't get a protein natively diffused into the coacervate. We have not seen a limit yet on protein that we put in as cargo. So, green fluorescent proteins are rather large molecules. It's not a colossal, 43K or KD or something. And that goes in just fine. Remember, RGD domain is just 161 amino acids at the end. So that's a rather large protein that we can get in. So that looks, so far we haven't seen a limit. I imagine there is going to be a limit, but we haven't seen it. Now, of course, if we did something like put MBP in between two RGD domains, that would not work, but MBP is colossal. So there's certain soluble proteins that I'm sure it won't work. But I can't say I could give you a definitive number, and I'm not sure it depends on the size per se. It could depend a lot on the chemistry of the protein. Mike Dustin, hi. When you make peptide-based bilayers, are they laterally mobile or do they have, okay. So that's a great question. So they're solid.
So hang on, can we finish?
I should really read it. Okay, okay. I'm sorry. Sorry. When you make the peptide-based bilayer, are they laterally mobile, or do you have the potential for solid LD and LO phases, as in phospholipid bilayers? What distinguishes cargo like luciferase from an inhibitor like MBP? Is it just size? Okay, I'll answer the first question first. So for the oleosin molecules, that's what we've been spending all of our time doing since the original PNAS publication, trying to soften the oleosin, because the oleosin 65s with the native hydrophobic core, has, I showed the structure of this. They have coil domains that strongly associate with each other. And there's like 6 threonine pairs in the interface, and that causes stabilization. And that leads to the membrane being basically a solid when we make it. So we're trying to soften the core with large extensive effort on trying to do mutations of the hydrophobic core in order to soften it. Because we'd love to have something that truly was liquid-like in our construction.
Can I just follow up on one thing?
Yeah, sure.
One additional, just on that before, I guess the other question may be related to the first one you answered also, but the, have you tried to do like this idea of having this very kind of strict hairpin could be interesting also like in a phospholipid bilayer for some applications that we run into with supported bilayers. So have you ever tried to put these oleosin molecules into a phospholipid bilayer is kind of like a reconstitution approach, or are they just too big?
We have tried, and we do have some evidence that they will go in. We didn't use 65s for that. We used 30s for that. So in other words, we trimmed down the hydrophobic core to 30 residues. And we did do this trick that I just described to you. We have this subset of oleosins, they're called 30Gs. So what we did was we mutated out all the threonines, so we have no hydrogen bonding. And then we replaced all the big bulky hydrophobic residues in the core with glycines. And those molecules will go into lipid membranes just fine. We've been using them for the project that I was talking to Carlise about at the beginning for making inhibitors to bind viruses. And they'll go in at some percentage into lipid membranes. But if you soften it enough, they'll go in as a dopant.
And do they stay monodispersed, I guess, in the bilayer, or do they kind of tend to cluster? I guess that's what you're trying to avoid by mutating out all
that we don't know. That we don't know if they cluster together into patches or not, or if they stay dispersed. We only did cryo-TM, and we know they're vesicular in structure, but we didn't do any imaging of the pair. I guess we could have done FRET pairs or something in order to figure out if we're forming clusters. I don't know that.
Yeah, thanks so much. It's really interesting. Thanks.
Oh yeah. As far as the second part of the question, I kind of muddled through the answer before. I think it's primarily based on size, but there's probably some chemistry effects as well. I don't think MBP would go in. I think it's too big. I don't know where the threshold is going to be though. Okay.
Great, thanks.
Great talk, Tosha. Hi, how are you? Great talk with huge potential. What would be the remaining challenges in this interdisciplinary work for real world applications? Thanks. Well, I'm a terrible person to ask about real world applications because I mean, there have been people who've done protocell migration in order to deliver drugs like in the brain. Battaglio's lab has done that. They were successful at doing that. You could do things like like motile systems interact with cells and make hybrid networks of cells and protocells, where protocells could act as hubs that could sequester and organize cells around them. That's one thing that we've thought about. I alluded to this before by saying one of the things we're interested in is when protocells move, do they move in environments with other cells? And what's the collective dynamics of how cells move? Do they move in streams, or they move in networks, or they move like snakes? Anna Balazs from Pittsburgh has done a lot of calculations suggesting how collections of particles might move. But I think we're very far from my lab, very far from using them to be used for, say, drug delivery per se. But I will say one thing which I did mention to Carlise right at the beginning before we started recording. So oleosin, this molecule oleosin that I was talking about before, there's lots of things you can do with oleosin. So one thing we have done, and we have a paper at ACS Nano showing this, we could put a binding peptides on the surface, on the head group of oleosin, that interfere with the binding between the spike protein of SARS-CoV-2 and the ACE2 receptor. And we made mice cells and other structures that have multiple copies of this binding inhibitory peptide. And that could be used to block the binding of the virus, the entry of the virus into host cell. And we've tested that with a variety of different strains of the virus, a lot of reporter viruses, and it seems to work wonderfully well. With very small concentrations of the protein. We think it works better than having the peptide by itself. The peptides we used came from the David Baker's lab, but ours seem to work a little bit better because they're multivalent, and that seems to add to the potency of the nanoparticles. So that would be the best real world application as an inhibitor of viral entry. Hopefully that will get acceptance.
Yeah. That's a wonderful application, but my question is more likely what would be the remaining challenges to go there?
Okay, so the remaining challenges to make an inhibitor, I think we've already dealt with that. We already made the structures. They seem to work very well. We're testing against live virus, and then we have to go into animal models in order to test in animals. As far as all the protocell work I was talking about before, I mean, we would like to have, what we're gearing toward is making a modular system where we could use any enzyme to drive the motion of a capsule over a surface, either randomly or directly. And we'd love to be able to control that from the inside out of a protocell. So first, the diversity of enzymes is very important. So we want to know, where are the limits to this? Which enzymes will work, which enzymes will not work? And then the issue is, could we get particles to respond to gradients? Could they change shape? Could they move through channels? In other words, how close to a motile cell could we get by making the formable protocell that could, say, squeeze into a junction? That's where we're headed with the protocell.
I see. Thank you so much. It's exciting. Really exciting. Thank you.
Thank you. That's the end of the questions in the chat. Any other questions? Any other live questions? You can ask a live question,
Dan, I just have one more thing I was curious about. So have you tried to, so I guess that core shell structure that you had in that last part is really interesting. If you link these was like RSS domains to a membrane, can you generate kind of like 2D coacervates that would where you could go from being in the whole membrane to kind of condensing into a 2D cluster? Or is this a system that only works in 3D because
I think that's a really good idea, and I've been kind of advancing this. There's actually a lab that's ahead of us on this. Gene Stokowiak's lab at Texas has been using transmembrane proteins with coacervating ends in order to put coacervates on the interior face of the cytoplasmic membrane, and that seems to be working pretty well. I would love to build a coacervate film on a surface. So, if you have little sticky groups on the surface of your coacervate, there's an interplay between the adhesion and the surface tension of the structure. I mean, I would imagine I could take a coacervate and spread it on a surface, and I'd love to make a motile coacervate. Is what we might ultimately be able to do with that.
Really interesting. Thanks. Thanks.
But you have complete control of the surfactant. You could put anything on the surfactant you want. You could put any adhesion molecule, since you're an adhesion molecule person. Any adhesion molecule or any ligand on the surface, and you can interact with receptors on a surface, and you can imagine all the things that one could do.
So I guess in the adhesion field, I guess there's some arguments about whether the everything that forms these 2D, what seem like phase separated systems qualify as liquid systems from a physical standpoint. I guess I guess you feel that these systems that you're working with, I guess they would meet all these criteria in terms of it's a liquid-liquid phase separation, and it's all the components within the structure. It's as opposed to like other physical phenomena with.
This is a really good question. So, and it gets to what is really going on inside of a coacervate. The coacervate has some structure at some length scale, but then it's a liquid at another length scale. If we do FRAP on the coacervate, it's purely liquid. Everything returns to its original shape. We can even measure the viscosity of it. But if I took a, let's say I could take a droplet, and I had stickers on the surface, and I put it on a substrate that was adhesive, and I could get it to spread, and I could get it to spread really, really thin. Then at some point, the liquid structure of the coacervate would obviously break down. It would no longer be a liquid film. I don't know how thin you have to make it before that's the case. But it's the kind of thing that could be tested with a system like we have, because we could actually get to a point, we could keep doing FRAP on spread liquids, spread coacervates on a surface to the point where we don't have a liquid structure anymore.
I wonder if also making it 2D could also cause it to lock up at some point, because maybe you'd potentially generate more structure or something. Yeah.
That's right. Right. You could induce, I can imagine inducing some kind of phase separation or a further phase separation on a surface by locking it up and putting little points down where you don't have the full freedom of the liquid in order to interrogate space. That all of that could happen.
Great. Thanks so much.
Yep. Oh, there's a question. Great talk. What is the pH of the buffer you typically use for encapsulation solution? The proteins that can use for making their vesicles should be dictated by some extent by that, right? Only certain proteins can be used based on the isoelectric point, perhaps. Great question. Okay, so we have it turns out fortuitously, the pK of the protein is like 7.4 or so. So that's about where the pH needs to be for the protein assembly. But this is a very prescient question, because if we were to start changing the amino acid sequence in the protein, we could change the pK, and then that would cause either changes in the assembly at a pH of 7.4, or would force us to go to pHs that are different in order to get the assembly to occur. So as you start to play with the sequence, you have to carefully monitor what the pK or the PI of the protein would be. But basically, we're fortunate that this is a naturally occurring protein, and we haven't changed the sequence very much, and we're about at neutral pH. Oh. Could you include other proteins inside the RGG phase? Yes. We've included many. For example, this is primarily Matt Good's work. So he just had a paper in Nature Chemical Biology where he showed that you could put the protein CDC24 inside of the RGG domain and then release it. And CDC24 is a cell cycle controller. So you could control the progression through the cell cycle by releasing CDC24 from the protein. And we have big plans to put lots of different functional proteins into the coacervates to control cell biology, both for motility and cell differentiation. So the answer is yes. The long answer is yes, Carlise.
This has been wonderful. I'm going to do a last call for questions before I stop the recording.
Well, it's great. It was my pleasure to give the talk. We had a thunderstorm roll in while the lecture was, I don't know if you heard the thunder, but that's why I kept looking over my shoulder like, oh my God, the house is going to blow away. So, but that would have been unfortunate.
Yes, indeed. I'm glad that didn't happen. All right, hang on. Let me make sure this is